Hello to all my dear students. I am Dipen Singh, your biology teacher. Welcome to all of you in our school SJEC e-learning YouTube channel. So student, in this biology class we discuss second unit, which is genetics and evolution's fifth chapter, which is principle of inheritance and variation. And so far in our previous video we already discussed the following terms related this chapter are heritance and variations, who is Mendel, the reason behind Mendel's success, and genetic terminology. And our today's topic to discuss is the deviation of Mendelism. The word Mendelism defined Gregor John Mendel, who suggests the remarkable laws related with the process of inheritance, means the transmission of genetically based character from one generation to another generation. And the term deviation related the concepts, the theories which are influenced by the three laws suggested by Mendel. Let's see. First of all, after the various discoveries about the Mendel's law, hereditary experiments were conducted on large number of animal and plants, and numerous exceptions to Mendelian ratios were observed, and these exceptions can be explained by suitably modified Mendel's law. And this type of discoveries, experiments, and ratios create or discover a new post Mendelian genetics, and it is also known as new Mendelian genetics, which classified into two types of interaction between gene. First type is interaction between the alleles of a gene. We can say that same gene, known as intragenic interaction or intra-allelic interaction. And the second interaction between the alleles of multiple genes, means when two alleles of a pair or more than two pair of alleles involve in the interaction known as intergenic. And we already know intra means the process between two individual of same species, while the intergenic means the two organism of different species is involved. The example related with intragenic interactions are incomplete dominance, when two alleles of a pair are not related as dominant and recessive. We say that incomplete dominance or partial dominance. Codominance means when both the alleles of a pair express themselves equally. Second, intergenic interactions examples are multiple genes, means there are more than two pair of genes responsible for a single factor or character. And the last is pleiotropism, means a multiple effect of a gene in an organism. First of all, we discuss about incomplete dominance or bleeding inheritance. As we know, in incomplete dominance, the genes of an allelomorphic pair are not related as dominant and recessive, but they express themselves when present together in the hybrid condition. Hybrid means when both characters are mixed together in a single group. As a result, the F1 hybrids exhibit a character intermediate to the effect of two parental alleles. Example can seen in Snapdragon. dog flower, or it is also known as Anthurium plant, present a good example of incomplete dominance when a cross is made between a true breeding red flower plant, which is denoted by capital R, and true breeding white flowered plant, which is represented by small r. Then the offspring, which were produced, all pink color flower, which is represented by capital R and small r in an hybrid condition. Let's see the cross. Here the cross showing incomplete dominance between pure red color plant, red color flower in plant, and pure white colored flower in plant. When the F1 pink flowered plants are self-pollinated or crossed among themselves, then F2 plants with red capital R, pink capital R and small r, and white flowers small r appeared in 1:2:1 ratio. What happened in this? Here the phenotypic ratio matches with the genotypic ratio of monohybrid cross, but the phenotypic ratio has changed from Mendelian dominant recessive ratio. And in F1 generation, capital R, which represent the red color, was not completely dominant over small r represented white color flower. So we can say that capital R and small r appeared as pink, and this is an intermediate expression of the two alleles. And we already use the cross method for F1 generation in this diagram, and Punnett square method to represent F2 generation in the example. First we take parent, which represent phenotype character as pure red and pure white, and genotypically character capital R and small r for red and white color respectively. Gametes obtained from the flowers are capital R for red color obtained from red color flower, small r for white color obtained from white colored flower, and they produce first generation or F1 generation, which are hybrid in their color orientation, and the hybrid color is pink, which is the mixture of red and white color. That's why this is known as hybrid pink. It means the red color is not completely dominant over white, and the hybrid represent there are some presence of white color character, and some also characters present represent red color. That's why the condition is incomplete dominance, which is carrying in the next generation cross represented by using the Punnett square, and the ratio obtained from this is 1:2:1, in which 1 represent pure red colored flower, 2 represent hybrid pink color flower, and the last one represent pure white color flower. Clear? Good. Let's see the second example of intergenic is both the genes of an allelomorphic pair express themselves equally in the F1 hybrid, means both character when share both types of alleles in a single group is known as codominance, and in F2 generation their phenotypic ratio matches with the genotypic ratio, the alleles of a pair which are able to express themselves independently when present together in a hybrid condition are called codominant alleles, and such case can be seen in coat color in cattle and in the case of blood groups found in human. Let's see first example, which is codominant factor of coat color in cattle. In cattle which carry gene R stand for red coat color and W for white coat color. When the red cattle, which represented by capital R crossed with white cattle represented by capital W, the F1 hybrids have roan color with genotype RW. The roan color is the mixture of red and white hairs which develop side by side in F1 heterozygous organism. Heterozygous organism means when the both characters are not same in an individuals. Both type of character are obtained from the different organism. And in F2 generation, red, roan and white cattle are produced in the ratio of 1:2:1 as can seen in the diagram. First, parental generation represented by the pure character, which is represented by red capital R and white capital W and capital W. By using crossing method, F1 generation is obtained and the color variation is changed. The color variation is roan color, which is the mixture of red and white. And when we obtain F2 generation, there is also some variation is obtained. We obtain pure red character and pure white character, and we also obtained the majority of roan color, which represent the codominant factor. Means in this roan case there are some character responsible for red color and also some character which are responsible for white color in the roan cattle. Let's see the second example of codominance of blood group. As we know, the human blood divided into two parts. 60 to 65% part of blood is made up of plasma, and remaining 35 to 40% part are made up of blood capsules. The capsules are divided into three types of cells: RBCs, WBCs and platelets. But here we are going to discuss about plasma membrane of human or plasma part of human. In the plasma membrane found in RBCs has sugar polymers which project out from the surface of RBCs, and on the basis of presence or absence of plasma membrane, the cells are denoted as antigen containing or absent of antigens. The type of sugar polymer formed is controlled by gene I, and the gene I has three alleles O, A and B, which are represented by IO, IA and IB respectively. Both the alleles IA and IB are dominant to allele IO but are themselves codominant also. In this case, alleles IO doesn't produce any surface antigen, while allele IA produces antigen A and allele IB produces antigen B. In every person, any two of the three alleles may be present. Therefore, the following types of genotypes and phenotypes are found in human population. Let's see in the diagram. Here is a diagram which represent blood types, their genotypes and also antigens. First of all, blood group O. Genotype responsible for blood group O represented by IO. As we already know, the alleles of a gene represented in pair. Antigen present in O is none, means there are no type of antigen present in blood group O, and the nature of O blood group is recessive, means these are the recessive character which express itself only the homozygous condition, while the blood group A represented by genotype IA in homozygous condition or IA and IO in heterozygous condition. Antigen A present in the blood group A and the nature responsible for blood group A is dominant. And same in the case of blood group B. The alleles responsible for genotype IB in homozygous condition or IB and IO represented in heterozygous condition. Antigen B present in blood group B which is dominant in nature. And the last pair of group which is AB group of blood, the genotypic ratio is IA and IB for both type of condition, homozygous and heterozygous. The antigen present is AB in the blood group B and the nature is codominant, and that is our topic. Codominant means both type of factors A and B both share equally in the F1 hybrids. That's why this factor is known as codominant factor. Our next and last example is pleiotropism. First of all, pleiotropism means what? It is the phenomenon of multiple effect of gene, means a single gene is responsible for multiple effect in a single organism, in which a single pair of genes influences more than one character at the same time. Such genes are called pleiotropic genes, and the genes produce more than one phenotypic effect which are totally unrelated. Let's correlate this explanation with Mendel, as Mendel proposed law of unit character, which means each character is controlled by one gene, a pleiotropic gene, along with controlling the manifestation of its phenotypic trait, and also influence some other related or unrelated character. Examples of pleiotropism are phenylketonuria and in Pisum sativum, the genes which control the shape of seed, round or wrinkled. Clear? First of all, we discuss about the example first, phenylketonuria. Phenylketonuria is an autosomal recessive character controlled by a mutant gene, and this mutant gene present on 12th chromosome of autosomal factor. The mutant gene, which is responsible for enzyme metabolism, code for the enzyme phenylalanine hydroxylase, also known as PAH, needed for the normal metabolism of amino acid phenylalanine to tyrosine. This result in the accumulation of phenylalanine in the body fluid like blood, sweat and cerebrospinal fluid, and appearance of an abnormal breakdown products phenylketones in urine. Accumulated phenylalanine and breakdown product cause severe brain damage leading to mental retardation, and such persons are fair skin, as we can see in the diagram, with blonde hair and blue eyes because of failure of melanin synthesis. Clear, student? So in our today's video we are just finished our topics. In next video we are going to discuss linkage and recombination. So thank you for watching our video, and if you like our way of teaching and hard work, please subscribe us to learn more and be updated about your syllabus.